In this video, we'll discuss some of the basics of sampling. One of the important things to realize about sampling is that no matter how accurate or how precise your analysis might be after the fact, if you don't have a good sample to start with, your analysis is complete junk. So in this video, we'll discuss three things. We'll spend a lot of time focusing on where within a target population you should collect your samples. The other thing is that we'll talk about what type of samples to collect and how many samples should be analyzed. So the first thing to consider about your sample is whether it's heterogeneous or homogeneous. The thing about most samples is that they're usually not homogeneous. If you picture the image off on the right here, we have different zones where you have different water quality. So where should we take our samples if we want to know how good this water is? Well, if you had a homogeneous sample, then it's okay to do one sample. But most samples are not homogeneous, and so that's no fun. So we have to think about what we mean by heterogeneity. The first thing to think about is the fact that it's not just space, okay? So in this picture here, of course, we have the zone of the good clean water, and we have the zone of some mucky water coming out from the river. But yeah, that's not spatially homogeneous. So with respect to being near the shore, where pollutants might be coming out or out from this river, you do have to consider differences going from left and right, up and down in a geographical zone. The other type of issue with heterogeneity is that things might change with time. That's called temporal heterogeneity. Classic example of this is the concentration of carbon dioxide. And on the curve on the right hand here, you'll see that there's kind of this zigzag going up and down. And what that is that the CO2 concentration is going to fall when we have the respiration of plants. So in the spring and summer, plants are using CO2 for photosynthesis. That's great. The CO2 concentration will decrease. And during the winter, when those plants are decomposing and not photosynthesizing, it'll increase again. And that's why you get this variation within an annual cycle. And there's all sorts of temporal heterogeneity. Blood glucose levels are going to change if you've just eaten a meal or not, for example. So we have to consider these. It is possible if you wanted to homogenize a sample. And so that means to mix it up. Usually this is impractical, especially when you're talking about environmental sampling. You can't just take that entire lake and mix it together and then take a piece. But if you had, say, a fruit or a bag of fruit, you could take those, put them in a blender, and mix them up and homogenize them. However, then you're losing possibly important details about variability within that bag. So let's just assume that your sample, like most of them, is heterogeneous, and so you want to go on and figure out which way to sample things. What a lot of people will fall back on is random sampling because they know that, hey, I want my sampling plan to be unbiased. And I know that random is totally unbiased. Well, here's the thing. Most people will do random just being like, oh, I'll pick from here and then there and then there. What that is haphazard sampling, and it's not truly a random plan. So pay attention now and learn about how random sampling actually should be done. If you want your sampling plan to be truly random, you have to first divide the population into subunits. That is done on the right hand here for a square sample, divided into 99 by 99 subunits. Okay. Second thing there is to assign a unique number to each unit, so like a row number and a column number together. This blue box, for example, could be 9801 if you want to do our row number. That's going to be 98 and then column number 01. All right, sweet. And then to make it actually random, you then have to have the random number table or a random number generator. And you'll, of course, restrict it to be between 0 and, I guess, 9999, because you can call this guy 9999. And so give me a random number between 0 and 9999. And if it comes up with 9801 as your random number generator, then that blue box is the one that you're sampling. You, of course, have to make multiple samples. But the key to this is divide it up, number the subunits, and then use a random number generator and random number table. Problem with this, although it is nice and unbiased, is that you usually have to make a lot of samples in order for this to give you a decent average of the overall population. And so more samples equals more time and more money, and usually you don't want to spend more time and money. So, judgmental sampling. This is where you pick something based on what you know about the sample. So you have some sort of previously determined information, or you have reasons for deciding for particular samples. A good example is that maybe you're trying to study weight loss. You want to see if a particular drug is going to help you lose weight over time. Well, you might want to get rid of patients who have other diseases. That might be a different factor. Okay. Judgmental, of course, is more biased, given that it's called judgmental, and that means you have a bias, compared to random sampling. But the advantage here is that you require fewer samples, and therefore you get to save time and save money. The third main type of sampling is systematic sampling. And in this case, you're sampling a population at regular intervals. And because your heterogeneity can occur in space, of course, you can have the intervals in space. So our image here of the lake, this is the Great Salt Lake in Utah. And apparently this is a satellite photo. You can find it in the Harvey chemistry textbook. And a railroad crosses the lake in the middle. You've got the portion above and below with different chemistries, and it's divided into a grid with regular intervals, and you take a sample in each of those grid locations at that regular interval. The other thing is that your intervals can occur in time, and so maybe something is changing periodically. The two graphs here are showing the signal, the real sample signal in blue, and then it's showing the number of data points that you're taking in red. And the thing here to recall is that if your data has a periodic trend, you can have a bias if you don't take your samples often enough. So the top one there, you're only taking a sample maybe once or twice per period of the actual signal. And you're not able to really get the proper frequency there. And on the bottom, you've got it nice. You've got multiple sample points, and you can actually describe what the frequency is of how this periodic signal is changing. The Nyquist theorem says that if you are going to collect a sample at a particular interval of delta t, so change in time, then the highest frequency, the fastest thing you can monitor correctly, is going to be 2 delta t, and then the inverse of that. To break this down, your signal period, suppose your signal period and the period again is like going from the same place, so from here to here is your signal period. If that's an hour, then you really should collect at least 3 or 4 points within the whole period, and so you should get your samples every 15 to 20 minutes. Now, we've talked about the 3 main approaches, random, judgmental, and systematic. You can combine these. The systematic-jugmental sampling plan is pretty common. For example, let's say we have our landfill, and the landfill is located right here at that dot. And you know that there's going to be the potential of waste leaching out of this landfill. You really want to prevent that. And so you're going to monitor that. Well, you know also that the groundwater is going to end up coming through in this direction right here. And so you're like, hey, groundwater is going that way. Lovely. We're going to monitor in the direction of groundwater flow. So that step, choosing the direction of analysis, is your judgmental portion. You don't want to monitor extensively upstream because your landfill shouldn't be affecting that. Well, maybe a point for reference, but you don't need to spend all the time and all the money analyzing the entire area around the landfill when you're really worried about what's happening downstream from the groundwater. Now that's your judgmental part. Systematic, you can see with that series of grids and the red dots is to break up that zone into the grid. So systematic, judgmental. Another type of combination is called stratified sampling. And what this really means is judgmental random. Imagine you want to analyze the caffeine or something in a bunch of Mountain Dew varieties. And as you can see, Mountain Dews come up with a whole rainbow of colors. And so you first have to divide that population of all Mountain Dew into strata or categories. And so the category here could be the brand or flavor. Then what you want to do is collect random samples within each stratum. So you're going to take the Mountain Dew code red, and you're going to go through and you're going to get, a whole bunch of bottles, lay them out in a grid, go through the steps of assigning numbers to each bottle, and then doing the random number generator to pick the bottles. Okay, so you still have to do the full out random sampling plan there within each stratum. Now you've analyzed each stratum or category, and so you're going to pool or average together the average values for each category, and that'll give you the overall population average. So the pro of this, why would you bother with this, is that within a particular category or stratum, you're going to have less variance than the overall population is. And so this will give you better reproducibility than just a full out random plan. The last part of picking a sample has nothing to do with minimizing error and variance, although I'm an analytical chemist, and so I think it's great to minimize error. But it really gets to the practical matter that sometimes you just need to do what's convenient. If you're out there and you're trying to figure out what's the quality of the groundwater, you are probably going to go and monitor it using existing groundwater wells like the one shown here. It's going to cost you a lot of money to divide a whole field into substrata or to go ahead and do a full-out random plan because you have to drill new wells. Convenience sampling is normally done to save time and money or because of accessibility, so I like to make fun of the possibility of you don't go into a store and be like, hey, does this skirt look good on me? To a random, like every 10th person that walks by, you're usually going to ask whoever happens to be in the fitting room with you at the point of time. Or if you were this guy, then you would ask all the people at your red carpet event. All right, so that's where to get the samples. A moment to talk about what type of sample. A grab sample is just a portion that you collect at a particular time or place. It's going to give you a snapshot of the composition. This is great because you can get individual variability, but it's really just, I grab it. You put the beaker in the lake and you pick it up. The composite sample is taking a bunch of grab samples and combining them together, thus making a composite mixture, and then analyzing that mixture. So if you want to know about the average composition, you can analyze just this one composite sample after you've mixed everything, instead of spending all the time and money to analyze, say, 10 separate grab samples and then average those results. The other thing is that a composite sample can really help you if a single grab sample can't give you enough material. If you want to look at a pollutant in an animal and the animal was small, like, I don't know, a worm or something like that, one worm isn't going to necessarily give you all of the mass that you need to get the accuracy that you require. The third option is to not take a sample at all. So grab and composite samples are removing a portion and taking it away to a lab. You could also do in situ monitoring, which is to monitor the population continuously with a sensor. Now, the last thing here involves a little bit of math, and it's the question of how much sample. And you've got the two ends of this issue. If you take too small of a sample, then the sample itself might be a different composition from your overall population that you're interested in. If you take too much sample, then you might be spending too much time and money without any additional information. So there's a formula here, and I'll help you define the terms. So N is the number of samples. So how many samples should I take? Okay. P is the probability of collecting the particular particle, for example, of interest. So let's just say you're interested in, I don't know, red M&Ms instead of all of the other M&Ms. Okay, so particle of interest. And then the other thing is how accurate and precise do you want to be? And this really comes down to precision. So here's our relative sampling variance right here. And let's just say that you want to analyze something that has two types of particles, particle A and particle B, okay? And you want your relative sampling variance to be 1%, so 1% error. And so this is going to be 0.01 because we have to put things in fractions. And let's just say that the thing that you're interested in is present at 1 part per billion. And 1 part per billion is going to lead to a P of 1 times 10 to the minus 9. Now, I'm not going to do it on the screen here, but if you plug in 1 times 10 to the minus 9 for each of the P values, if you plug in 0.01 here for your relative sampling error, what N comes out to be is N is going to be 1 times 10 to the 13. You need 1 times 10 to the 13, which is a huge exponent, particles in order to get 1% sampling error on something that's 1 part per billion. And it is fairly common that in analytical chemistry you do have something that is that trace of a concentration. Now, if it's a solid and you have particles, then 1 times 10 to the 13 can really be a huge mass. And mass is not actually the factor here. The factor here is the number of particles. So if you want more particles for less mass, what you do is you grind up your sample. You're going to use that mortar pestle, and you're going to reduce the particle size, therefore increasing the number of particles that are available to you in a given mass. So I hope that was a helpful introduction to sampling, and I look forward to seeing you at future videos.